Bismillahirrahmanirrahim. Assalamu alaikum. My topic name is Nano drugs for skin cancer. Table of content. Firstly, we discuss the table of content, introduction, background on skin cancer, current treatment approaches and challenges. The need for innovative solutions, nanotechnology revolutionizing cancer treatment, benefits and application in cancer treatment, drug nanoparticles enhancing cancer treatment, mechanisms of drug nanoparticle action in cancer treatment, formulation and synthesis of drug nanoparticles, evaluation of drug nanoparticles, future direction and challenges. Introduction. Skin cancer is a significant public health concern that affects millions of people worldwide. In this presentation, we will delve into the prevalence and types of skin cancer, including melanoma and non-melanoma skin cancer. Additionally, we will explore the current treatment approaches and challenges associated with them, highlighting the pressing need for innovative solutions. Background on skin cancer. Skin cancer is a abnormal growth of skin cells typically caused by excessive exposure to the ultraviolet radiation from the sun or artificial sources such as tanning beds. It is a most common form of the cancer globally, with its incidence steadily rising over the past few decades. There are primarily the three types of skin cancer: melanoma, squamous cell carcinoma, and basal cell carcinoma. Melanoma is a most aggressive, deadliest form of skin cancer. It helps from the melanocytes, pigments producing cells in the skin. Although it accounts for a smaller percentage of skin cancer cases, it is responsible for the majority of skin cancer related deaths. Number 2, non-melanoma skin cancer. Non-melanoma skin cancer, namely squamous cell carcinoma and basal cell carcinoma, are most common but less aggressive than melanoma. Squamous cell carcinoma arises from the squamous cells, while the basal cell carcinoma originates in the basal cell of the skin. While these types of skin cancer are generally less lethal than melanoma, they still require prompt diagnosis and treatment. Current treatment approaches and challenges. Traditional treatments for skin cancer including surgery, chemotherapy and radiation therapy, but they have limitations. Surgery, it involves removing the cancerous growth, but extensive surgery can result in disfigurement, especially in sensitive areas. Chemotherapy used for advanced skin cancer, it is often has a severe side effects and some cancer may not respond well. Radiation therapy destroys cancer cells but can damage surrounding healthy tissues, leading to long-term effects. The need for innovative solutions. Innovative solutions are needed to overcome these limitations and improve outcomes for skin cancer patients. Targeted therapies, developing treatments that specifically target cancer cell while minimizing damage to healthy cells. Immunotherapy, enhancing the body immune system to recognize and eliminate cancer cell. Photodynamic therapy, using light and photosensitizing agents to selectively destroy cancer cells. Nanotechnology based approaches, utilizing nanoparticles for precise drug delivery and improved treatment efficiency. Nanotechnology revolutionizing cancer treatment. Nanotechnology, manipulation of matter at the nanoscale, has emerged as a groundbreaking field with vast implications across various disciplines. In cancer treatment, nanotechnology has revolutionized the way we approach the disease, offering unprecedented opportunities for targeted drug delivery, improved imaging techniques, and early detection methods. Today, we will explore the significance of nanotechnology in cancer treatment and its diverse applications. Overview of nanotechnology. Nanotechnology involves the manipulation and control of materials at the nanoscale, typically ranging from 1 to 100 nanometers. At this scale, material exhibit unique properties and behaviors that differ from their bulk counterparts. Nanotechnology has found application in fields such as electronics, material science, energy, medicine. Its ability to engineer materials at such as small scale opens up new possibilities for addressing complex challenges, including cancer treatment. Benefits and application in cancer treatment. Nanotechnology has transformed cancer treatment for providing the innovative solution to overcome the limitation of conventional therapies. Here are key benefits and application of nanotechnology in the field of cancer treatment. Targeted drug delivery. Nanoparticles can be designed to specifically target cancer cells, delivering therapeutic agents directly to the tumor site. These targeted approaches improve drug efficacy while minimizing toxicity to healthy tissues, reducing side effects and enhancing patient outcomes. Improved imaging techniques. Nanoparticles can be utilized as contrast agents in medical imaging, enhancing the detection and visualization of tumors. These imaging techniques, such as nanoparticle enhanced MRI or CT scans, allow for more accurate staging and monitoring of cancer progression, enabling timely interventions. Early detection method. Nanotechnology based approaches offer the potential for early cancer detection through sensitive and specific biomarker detections. Nanosensor and nano devices can detect and analyze new tumor specific biomarker in body fluids, facilitating early diagnosis intervention when the disease is most treatable. Enhance the therapeutic efficacy. Nanotechnology based offer the potential for early cancer detection through the sensitive and specific biomarker detection. Nanosensor and nano devices can detect and analyze a tumor specific biomarker in body fluid, facilitating early diagnosis intervention when the disease is most treatable. Enhance the therapeutic efficacy. Nanoparticles can be engineered to carry multiple therapeutic agents or combination therapies, in improving treatment efficacy and overcoming drug resistance. Additionally, their unique property enable controlled release of drugs, ensuring the sustained drug level and minimizing frequent dosing. Drug nanoparticles enhancing cancer treatment. Drug nanoparticles play a crucial role in advancing cancer treatment. These tiny particles exhibit the unique property that enable targeted drug delivery and improve the therapeutic outcomes. In this section, we will define the drug nanoparticle, explore their characteristics, and discuss their different types used in the cancer treatment. Definition and characteristics of the drug nanoparticles. Drugs nanoparticles refer to submicrons size particles engineered to deliver therapeutic agent to specific site in the body. Their unique property include: number 1, size. Drug nanoparticles typically range from 1 to 100 nanometers in diameters, allowing them to interact with the cell and tissues at a molecular level. Number 2, shape. Nanoparticles can be engineered into various shapes such as spheres, rods, or tubes, which influence their behavior, stability, and cellular uptake. Number 3, surface area to volume ratio. Drug nanoparticles have a high surface area to volume ratio, enabling efficient drug loading and increased interaction with targeted cells. Chemophysical Characteristics of nanoparticles employed for drug delivery in skin cancer. In cancer treatment, various types of drug nanoparticles have been developed and utilized to enhance the delivery, targeting, and efficacy of anti-cancer drugs. Here are some commonly used types of drug nanoparticle along with their details. Number 1, liposomes. Liposomes are spherical vesicle composed of lipids bilayer. They are widely used in cancer treatment due to their biocompatibility, versatility, and ability to encapsulate both hydrophilic and hydrophobic drugs. Liposomes can be modified to improve stability, circulation time, and targeting specificity. They can passively accumulate in tumors due to enhance the permeability and retention effect. Polymeric nano particles. Polymeric nanoparticles are composed of synthetic and neutral polymers. They offer flexibility in drug encapsulation, surface modification, and controlled release. Polymeric nanoparticles can improve drug solubility, stability, and bioavailability. Common polymer used include the polylacticoglycolic acid, PLG, polyethylene glycol, and chitosan. Number 3, dendrimers. Dendrimers are a highly branched nanoparticle with a well-defined structure. They offer precise control over size, surface functionality, and drug loading capacity. Dendrimers can encapsulate drugs within their interior or conjugate them on their surface. Their compact size and tunable property make them attractive for targeted drug delivery and imaging application. Number 4, nanocrystals. Nanocrystal consist of drug molecule in crystalline form at the nanoscale. They enhance drug dye dissolutions and bioavailability, particularly for poorly soluble drugs. Nanocrystals can be surface modified to improve stability and target specific tissue. They are often administered via injection or as a part of topical formulation. Number 4, gold nanoparticles. Gold nanoparticle possesses unique optical and physiochemical properties. They can functionalize with drugs or targeting ligand to for site-specific drug delivery. Gold nanoparticles are used in cancer treatment for imaging, photothermal therapy, and as a carrier for drug delivery systems. Number 6, magnetic nanoparticles. Magnetic nanoparticles such as the iron oxide nanoparticle are equipped with magnetic property that enable their manipulation and targeting using the external magnetic field. They can be loaded with drugs and guided to tumor site, allowing for localized drug delivery and hyperthermia therapy. Number 7, carbon nanotubes. Carbon nanotubes are a cylindrical structure composed of carbon atoms. They have a high drug loading capacity, can be functionalized for targeted drug delivery. Carbon nanotubes show promise in photothermal therapy and imaging application. Number 8, hybrid nanoparticles. Hybrid nanoparticles combine different types of materials such as lipid, polymers, metals, or inorganic materials to leverage their unique properties. These nanoparticles can offer multifunctionality, improve drug loading, and control release profiles. They are often designed to achieve targeted drug delivery, imaging, and therapeutic synergies. Mechanism of drug nanoparticle action in cancer treatment. Drug nanoparticles offer unique mechanism of action in cancer treatment, enabling targeted drug delivery while minimizing damage to the healthy cells. In this section, we will explore how drug nanoparticle achieve selectively targeting through their active and passive strategies, as well as the enhance the permission and retention effect. Targeted drug delivery. Drug delivery nanoparticles are selectively target cancer cells, maximizing therapeutic efficacy and minimizing side effects on the healthy tissues. The following mechanism contribute to their targeted drug delivery. Number 1, enhance the permission and retention effect. The EPIR effect is a phenomena observed in tumor tissue characterized by leaky blood vessels and impaired lymphatic drainage. During the nanoparticles exploit this effect by accumulating preferentially in tumor tissues while sparingly healthy tissues. Active targeting involves the attaching specific ligands such as antibodies or peptides to the surface of drug nanoparticle. These ligands can recognize and bind to the receptor that are overexpressed on the surface of cancer cell, facilitating selective uptake and internationalization of nanoparticles into cancer cells. Passive targeting. Passive targeting takes advantages of the EPIR effect mentioned earlier. Drug nanoparticles, due to their small size and specific surface properties, can passively accumulate in tumor tissues through the leaky blood vessel. This accumulation is a more pronounced in tumors compared to the healthy tissues, resulting in the preferential drug delivery to cancer cells. Formulation and synthesis of drug nanoparticles. The formulation and synthesis of drugs nanoparticles play a critical role in their effectiveness as a therapeutic agents in cancer treatment. In this section, we will explore the common method used for synthesizing drug nanoparticles, the factor influencing their formulation, and specific example of nanoparticle formulation for skin cancer treatment. Method for drugs nanoparticle synthesis. Several methods are used for synthesizing drug nanoparticles, include nanoprecipitation. This method involves the rapid mixing of a drug and polymers solutions resulting in the precipitation of nanoparticles. Nanoprecipitation is a simple, cost-effective and suitable for a wide range of drugs and polymers. Number 2, emulsion solvent evaporation. In this method, the drug is dissolved in water immiscible organic solvent, which is then emulsified with the aqueous phase containing a stabilizer. The organic solvent is subsequently evaporated, leading to the formation of drug nanoparticles. Self-assembly technique. Self-assembly involves the spontaneous organization of drug molecules or polymers into nanoparticles. This can be achieved through methods such as the conservation, metallization, or nano-emulsion techniques. Factor influencing nanoparticle formulation. Several factors influence the formulation of nanoparticles, including number 1, drug loading. The amount of drug that can be loaded into the nanoparticles affect their therapeutic efficacy. Optimizing the drug loading is crucial to ensure the effective drug delivery. Number 2, stability. Nanoparticles should remain stable during the storage and circulation to maintain their integrity and drug release properties. Factors such as pH, temperature, interaction with biological components must be considered for achieving long-term stability. Number 3, surface modification. Surface modification of nanoparticles can enhance their stability, prolong circulation time, and enable specific targeting. Coating nanoparticles with ligands or polymers can influence their interaction with cells and tissues. Number 4, release kinetics. The release of drugs from nanoparticles should be controlled to ensure the sustained drug levels at the targeted site. Release kinetics can be modulated through the factors such as nanoparticles, composition, size, and surface properties. Example of nanoparticle formulation of skin cancer. In the skin cancer treatment, specific examples of drug nanoparticle formulation including number 1, liposomal doxorubicin. Liposomal loaded with doxorubicin, a chemotherapy drug, have been developed to improve its targeting and reduce the toxicity. Number 2, polymeric nanoparticles. Polymeric nanoparticles such as PLGA nanoparticles have been used to encapsulate anti-cancer drug like paclitaxel for targeted delivery to skin cancer cells. Number 3, metallic nanoparticles. Gold nanoparticle functionalized with drugs or antibodies have shown potential for both targeted drug delivery and photothermal therapy in skin cancer. Evaluation of drugs nanoparticles, ensuring efficacy and safety. The evaluation of drug nanoparticles is crucial to ensure their efficacy and safety in cancer treatment. In this section, we will discuss the characterization techniques used to assess the properties of drug nanoparticles, the method for evaluating their efficacy in vitro and in vivo, and the importance of the assessing safety and toxicity consideration. Characterization techniques for drug nanoparticles. Several techniques are used to characterize drug nanoparticles, including number one, dynamic light scattering. Dynamic light scattering measures the size, distribution, and stability of nanoparticle in a solution, providing information on their hydrodynamic diameters and aggregation behavior. Number three, transmission electron microscopy. TEM allows for visualizing of nanoparticles at a high resolution, providing the information about their size, shape, and morphology. Fourier transform infrared spectroscopy. FTIR analyzes the chemical composition of nanoparticles by measuring the absorption and transmission of infrared light, enabling the identification of functional group and surface modification. In vitro and vivo evaluation method. Various methods are employed to evaluate the efficacy and safety of drug nanoparticles. Number one, cell viability assays. In vitro cell viability assays such as MTT and or ALMAR blue assays the cytotoxic effects of drug nanoparticles on cancer cells, determining their anti-cancer activity. Number two, drug release studies. These studies measure the release kinetics of drugs from nanoparticle, providing information on their controlled drug release behavior and stability over the time. Number three, animal models. In vivo studies using animal models help evaluate the therapeutic efficacy, biodistribution, and potential toxicity of the drug nanoparticle in a complex biological system. Safety and toxicity consideration. Assesses the safety profiles and potential toxicity of drug nanoparticle is of the utmost importance. Biocompatibility. Evaluation. The biocompatibility of nanoparticles include assessing their interaction with cells, tissue, or biological barriers to ensure the minimal adverse effects. Number two, immunotoxicity. Assessing the immunotoxicity of a drug nanoparticles involve examining their effects on the immune system, including immune cell activation, cytokine release, and potential immunogenicity. Number three, systemic toxicity. Evaluating systematic toxicity involves the monitoring the impact of drug nanoparticle on the vital organs, hematological parameters, and overall physiological function. Future direction and challenges. Future direction and challenges in drug nanoparticles research. The field of drug nanoparticle research is rapidly evolving, offering exciting prospects for future of cancer treatment. In this section, we will discuss emerging trends and future direction in the drug nanoparticle research, with a particular focus on the combination therapies and personalized medicine. We will also address the challenges that need to be overcome to fully realize the potential of drug nanoparticles in clinical practice. Emerging trends and future prospects. Number one, combination therapies. One, emerging trends is the use of drug nanoparticles in combination therapies. There are multiple therapeutic agents are delivered simultaneously or sequentially. This approach can target multiple pathways, overcome the drug resistance, enhance the treatment outcome. Number two, personalized medicine. The development of personalized medicine approaches is gaining momentum by tailoring drug nanoparticles to individual patients based on their unique tumors, characteristics, genetic profiles, and therapeutic needs. More precise and effective treatment can be delivered. Number three, multifunctional nanoparticles. Researchers are exploring the design of multifunctional nanoparticles that can carry multiple payloads, including drugs, imaging agents, and targeting ligands. These nanoparticles can enable simultaneously diagnosis, treatment, and monitoring of cancer, leading to improve the patient management. Challenges. While drug nanoparticles hold great promise, several challenges need to be addressed. Number one, regulatory hurdles. Navigation regulatory requirement for clinical translation and approval of drug nanoparticles can be complex. Streamlining these processes is necessary to ensure timely access to innovative therapies. Number two, manufacturing scalability. Ensure the scalable and cost effective production of drug nanoparticle while maintaining their quality and consistency is the challenge that need to be overcome for wide spread adoption. Number three, long-term safety and toxicity. Comprehensive long-term safety assessments are essential to ensure the safety and minimize potential toxic effects of drugs nanoparticle over extended treatment periods. Conclusion. The future of nanoparticle research in cancer treatment is promising with the emerging trends focusing on combination therapies, personalized medicines, multifunctional nanoparticles, overcoming challenges related to regulatory processes, manufacturing scalability, and long-term safety assessments is a crucial for successful translation of drug nanoparticle into clinical practice. By addressing these challenges, we can unlock the full potential of drug nanoparticles and revolutionize the field of cancer treatment, offering new hope and improved outcomes for patients. Thank you for listening.